I have some plans for today. So we're going to start talking about how we cultivate microbes. And we're going to start with the very first step. There are so-called 5 I's: inoculation, incubation, isolation, inspection, and identification. So I kind of hope to get to inspection, I'm sorry, but we'll see how it goes. So let's start with the first I, which is inoculation. And the key thing here is that inoculation process should be sterile, okay? What do I mean by that? Today you used the cotton swab. It was sterile. TSA plates, suborod plates were sterile, okay? So you don't have any contamination of the sample, right? Sterile collection technique, swab, sterile medium. This brings us to the concept of medium. So what is the medium in microbiology? Medium is anything that supports the growth of microbes. TSA, trypticase soy agar, medium. Milk, medium. Chicken stock, medium. Mice, medium. We are a medium. Does that, you can consider us medium for microbes. All right, so how do we classify them? And I'm going to highlight some features. So if you want to classify medium or media by physical state, solid medium, agar or gelatin, okay? The feature of this medium is that it's liquefiable. What does that mean? You heat it up, it becomes liquid. Cool it down, it becomes solid. Are we clear? It's convenient because you can sterilize it, pour it in the plate, it will solidify in a nice little layer of agar. What is it suitable for? Isolation of microbes, growing them in the form of colonies. Selection of microbes, you can use medium that supports the growth of one microbe but not another. And you can differentiate microbes using the medium. On another end of the spectrum here are liquid media. So liquid media, well, obviously liquid, okay? Sometimes they're called broth. Okay, so chicken broth is medium. They're really good for, let's call it preparatory growth. What I mean by that? If you need to accumulate, I don't know, 5 pounds of microbes, that's the way to go. You see what I'm saying? You can throw them in the, you can throw the medium in the tank and just grow them in massive amounts. Liquid medium is no good for isolation. If you have 2 microbial species growing in the liquid medium, you can't separate them. You can't tell them apart. Between the solid and liquid is semi-solid. It looks like a clot, really. Okay, there's no solid surface. It's not liquid though. So if you flip the tube with a semi-solid medium, eventually it will get out of the tube, but it'll take days. Okay. Let's call it broadly characterization. You can use it to characterize organisms like whether they aerobic or anaerobic, whether they have capsule or don't have capsule, whether they're motile, non-motile, fermenting, non-fermenting. There's like a ton of things that can be done with semi-solid medium. It's not really good for isolation. It's not really good for preparatory growth. Okay? Make sense? Right, chemical composition. Two types. Chemically defined medium and chemically complex medium. Now, I'm not going to put a lot of stuff on the board, but I'm going to discuss with you what's the difference here. So, what is chemically defined medium? Imagine that you lined up your 21 amino acids, some sugars like glucose, sodium chloride, I don't know, potassium chloride, and water, right? And you measured the exact amount of each component. Are you following me? And you put it in the exact amount of water, so at the end of the day, you know the exact chemical composition of your medium. You see what I'm saying? Exact chemical composition. So what's the difference between defined, that was the description of defined medium, and the complex. So your trypticase soy agar is the complex medium. You take soy protein, treat it with a trypsin, break it down in pieces, throw it in the water with some salt and stuff. Do you know the exact chemical composition of that digestion? No. You see what I'm saying? Like chicken stock is complex medium. You don't know the exact chemical composition. Brain heart infusion, you just take brain, heart, dry them, grind them, put them in a water. You don't know what's there. You know that there's a lot of nutrients, right? So complex medium is really good to grow a huge number of different microbes, because you know for sure there are plenty of nutrients. What's the goal of defined medium? Let's say you want to study whether a particular strain of microorganisms requires histidine, amino acid histidine, to grow. You see what I'm saying? So you can remove histidine and see if they still grow. And then add histidine and see if they grow. Like this is your controlled variables here. Okay? Good? Now, finally, the classification of media based on purpose. So you have general purpose medium, TSA agar, just garden variety. Most of the stuff will grow. Nothing special about it. Then you have so-called enriched media. Okay, so what is enriched medium? There are some microorganisms that are called fastidious, or in normal people's words, picky. For instance, remember we discussed not UTI but STI, okay? So Neisseria gonorrhea is a fastidious microorganism that requires a lot of iron. So you can only grow it on the enriched medium such as blood agar or chocolate agar. Both of them contain blood in one or another form, meaning they contain a lot of iron. That makes sense? So, then you have selective medium. That's your Saborot agar. It inhibits the growth of bacteria, but supports the growth of fungi. Or McConkie agar inhibits the growth of gram-positives, but supports the growth of gram-negatives. Do you get the gist of selective? So it selects for something. And finally, differential medium allows you to differentiate different microorganisms based on certain feature. So let's talk about, give as an example, let's use McConkie agar. So if you grow microorganisms on McConkie agar, is it selective? Yes, it will select for gram-negatives. Are you with me? It will also allow you to distinguish between lactose fermenting and lactose non-fermenting bacteria. So you've got selective and differential. See what I'm saying? So what you can expect in terms of questions here. I give you a description, a real life description of the medium. Like blah, 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 McConkie agar. You tell me, okay, McConkie agar, solid. It inhibits the growth of gram-positives, selective. Allows you to distinguish lactose fermenters, non-fermenters, differential. you find the line that states solid selective differential medium, pick it, that's your answer. Does that make sense? Matching the description with a proper set of terms. Okay?
So differential is selective and differential.
Differential is blood. So those are two independent terms.
Okay.
Differential doesn't have to be selective.
Okay.
Thank you for the question, by the way. Say there is a medium called chromagar. It's a clinically used medium that allows you to distinguish between various urinary tract pathogens. They will grow in different colors. It will be Staph aureus, Klebsiella, E. coli, Proteus mirabilis, Staph saprophyticus. They're all gonna grow, they're all different, but they all look different. So differential medium supports the growth of a lot of microbes, but allows you to distinguish between them.
So the
Distinguishing part
okay.
is because we call it differential.
Okay.
Does that make sense?
Yeah, so then how can it be both of them? Like the example you just did?
Yeah, so if the medium restricts the growth of some, but the ones that are allowed will look different because of a certain feature, it's both selective and differential. That makes sense? Okay, good. Couple more words about media. So types of culture. Not even going to put. There are 3 types. In lecture, just know the definitions. Azenic is the pure culture, 1 species. Mixed culture, many species. Contaminated culture, you were hoping for 1, but screwed up, and now you have a contaminant. Does that make sense? It's like mixed culture is when you accepting everyone. Contaminated culture, when you didn't want that contaminant to be there, right? Make sense?